He's founded the Living Earth Collaborative there in St. Louis, and that's a partnership between Washington University, the St. Louis Zoo, and Missouri Botanical Garden. And they are bringing together all the top biodiversity researchers in a collaborative to address the impacts on biodiversity and the challenges facing the Earth in sustaining life. Jonathan, as you may know, has had a remarkable research track record. He's got 2 books, Improbable Destinies, that you may have recently seen. I think it came out last year. Improbable Destinies, Fate, Chance, and the Future of Evolution. And then the book that came out a few years back, Lizards in an Evolutionary Tree, and that's all about Anoles. He's also the author on several textbooks. He has more than 350 research papers to his name. Many of these have been published in top journals such as Nature, Science, PNAS, Evolution, and many others. He's a member of the National Academy of Sciences. He's received the Dobansky Prize and the E.O. Wilson Naturalist Award and the David Starr Jordan Prize. So we are very, very pleased to have him here today with us, with the HAA, doing a Zoom webinar for us on his favorite topic, I believe, the Anoles. Jonathan, thanks. I think you can start if you wish, yeah.
Thank you, Crystal and John. I'm very flattered to be the guinea pig for these seminars and to be asked to speak to you. And it's a delight to be addressing the Herpetological Association of Africa again. I attended several meetings many years ago and gave one lecture. So I'm delighted to be back and to talking to you today. And I should say good afternoon to most of you in the afternoon. It's morning here in St. Louis in the middle of the United States. So there we go. Here's my title. And we're going to start when I was back in university as a college student. And this is me as a younger man. You can see, well, you can see I'm older now. And I remember very clearly when I was a third-year student, a junior, attending a lecture in the biology department's lecture series by John Endler. It was on his work on natural selection on guppies. And that work to this day, I remember very well. Endler had gone to Trinidad, where guppies are native, and he had found something, actually someone else had discovered it first, but he confirmed it, that there was great variation among guppies in how colorful they are. Some of them look like this guppy here, with spots and stripes and colors, but others are quite bland in coloration. I should point out, these are the males that vary so greatly. All females are bland. And he found that there was something associated with the variation in color, and that was the presence of larger predatory fish. That in the presence of predators, they're bland, in the absence, they're colorful. Well, this association suggested a causal relationship, and the idea was that females prefer brighter males, something we now know to be true. Why that is, we don't know, but females just love a flashy male. And in the absence of predators, there's strong sexual selection for males to get colorful. But in the presence of predators, they can't afford to be colorful because the predatory fish pick them off. So that was the idea, but of course, that was just a correlation, and we all know correlation does not prove causation. And so Endler, and remember, this is back in the late 70s, decided to use the powerhouse of modern science, the experiment, to actually test this idea, something that was very radical at the time in evolutionary biology. And what he did is he went to a little pond or a little pool in the mountains where both species occurred and where the guppies were bland in color. And he simply moved the guppies higher up in the stream to where the predators didn't occur. And his idea was that if in fact the predators were responsible for the lack of coloration, then when you move the guppies to a predatorless area, they should evolve to be more colorful. And sure enough, that's exactly what happened. In a relatively short period of time, just a few guppy generations, they evolved to become much more powerful. More studies in greenhouse, in large pools and greenhouses showed the same thing. Well, this was a stunning experimental demonstration. And what it showed is that we can test ideas about evolution experimentally. Now, at the same time that this work was being done on the guppies, it was the same time that the famous Princeton biologists, Peter and Rosemary Grant, were doing their work showing rapid evolution on Darwin's finches. And so we could see that evolution occurs rapidly. Now, the context for this work is we had known that when natural selection is strong, evolution occurs rapidly. We knew that from decades of studies on fruit flies, where if you selected on a trait, very quickly the population would evolve in the direction of selection. And we knew from nature, the famous example of the peppered moth, that when pollution covered the trees, the moths evolved to become darker in color very quickly. And conversely, when pollution was removed, they evolved back to their original peppered coloration. And in fact, we've also seen rapid evolution as species respond to how we're messing with the environment. The evolution of pesticides, of herbicides, of insecticides, all of these are examples of populations evolving quickly when natural selection pressures change. And of course, the evolution of antibiotic resistance. What was new, though, about the guppies and about the finches was that this was occurring out in nature in relatively normal conditions. One could argue that the selection pressures from pesticides and so on, and from pollution, were unnaturally strong, that humans had messed up the environment. What Endler's work and LaGrance's work showed that even in seemingly natural situations, natural selection pressures could be strong, evolution could occur very quickly. And as Endler's work showed, you could actually do experiments in evolution and hope to get results within a reasonable period of time. Well, that was the message I got from the talk of Endler's that I saw close to 40 years ago now. And that resonated with me. So let me give you the outline of the talk today. It's going to be experimental field studies of evolution in the Bahamas. And this will be a play in 4 acts. There we go. We'll first talk about adaptation to different environments, then adaptation to interspecific competition, then predation, finally a little talk about founder effects, and if there's time, an encore with some new work. So I'll be speaking a little bit quickly because I want to get to that encore. So let's start with part 1. First, I need to recognize that this work has been collaborative from the very outset. I've worked with many different people, but the main players in this work, first is Tom Shaner, the eminent ecologist from the University of California, Davis. He was my postdoctoral mentor, and I've worked with him ever since. In addition, Dave Spiller, who's worked with Shaner for many years, was also a key part. Much of the later work were 2 people who started out as graduate students in my lab and are now faculty in their own right, Manuel Leal at the University of Missouri and Jason Colby at the University of Rhode Island. So let's get on with the show. Act 1, adaptation to different environments. And I need to introduce now our one other actor, and that is the star of the show, this lizard, Anoles sagerii, the brown anole. Now, probably you're wondering, why is somebody who does his research in the Caribbean talking to the Herpetological Association of Africa? Well, you'd have to ask Crystal and John that, but there is a slight relevance, and here it is. Anoles sagerii. Anoles is the largest genus of vertebrates, of amniotes in the world. There are 400 recognized species throughout the New World tropical and subtropical areas. Of these species, the brown anole has the largest distribution. Here is its natural distribution in the Caribbean along the coast of Central America. So it has a very broad distribution, but it's also been introduced. So that's just to place it in the world where the Caribbean is, in case I realize we're halfway around the world from most of you. But the brown anole turns out to be a great invader, and it has shown up in all kinds of places, elsewhere in the United States, in Mexico, Panama and Costa Rica. It's now in South America, in Ecuador. Over in Asia, it's well established both in Taiwan and in Singapore. It's in Europe and Spain. So it's hit most of the continents. And I should tell you, and here is your relevance, the brown anole is now in Africa. Aaron Bauer and Luis Seriaco are reporting the brown anole from Angola. So that's not very far from where many of you are. And the brown anole, being a great invader, who knows how far it will spread in Africa and throughout the world. So perhaps there's a slight bit of relevance in this talk, the brown anole coming soon to a neighborhood near you. Incidentally, the primary means it gets moved around seems to be in potted plants, in the nursery trade. As people move around ornamental plants or plants for planting in gardens, the lizards sneak away, or they're eggs, and that's how they get moved around. All right. Well, my doctoral work, going back to my time as a PhD student at Berkeley, was on the phylogenetic relationships of anoles and the evolution of their characteristics. And in particular, one feature that I looked at was how long their legs are. And there's variation between very long-legged lizards here on the left, perhaps slightly reminiscent to tree agamas, although much smaller, but long legs living on broad surfaces, and short-legged species using narrow surfaces we call twig anoles. And this dichotomy evolved many times within anoles. The evolution repeatedly of twig anoles using narrow surfaces and of species with long legs on broad surfaces. And functional studies we did on how fast they can run and so on provided a biomechanistic explanation for this correlation, that long legs allow a lizard to run faster. They can take longer strides, and on broad surfaces, they're very beneficial, presumably. Whereas on narrow surfaces, short legs are much better for dexterity, for keeping from moving carefully and not falling off the surfaces. And so we seem to understand the cause for this convergent evolution. But again, this is a correlation, and it certainly makes sense, but we don't see the causal connection between the two. That wasn't established. Anyway, that was my doctoral work. Well, as a graduate student, I read this paper by Tom Shaner and his then wife, Amy, where they put brown anoles on very tiny islands in the Bahamas. And the reason they did that is that they found that naturally the brown anole did not occur on these islands, and they wanted to study extinction. And so they put a small number of lizards on these islands, expecting the populations to go extinct. They would watch what happened and learn something about the process of extinction, something we still need to know more about today. So here is where the study was being conducted. It's in the middle of the Bahamas, around a place called Staniel Key. Here's Florida and Cuba to give you some bearings. Here are the islands. Here's some of them right here in the middle, these small ones. I hope you can see the arrow on my screen. Here's a map. Look at the legend here. That's a kilometer. So these are small islands, tiny islands, islands the size of, well, I compare them to baseball diamonds, but that probably doesn't mean much to most of you, maybe half of a basketball court. So they're very small islands. And so the Shaners put 5 or 10 brown anoles on these islands, expected them to go extinct, and in fact, almost none of them did. This graph shows how long it took them to go extinct as a function of the size of the island. And what you can see is that islands slightly bigger than 10 square meters, a very tiny island, the populations survived indefinitely. In other words, put lizards on these islands, unless they're completely tiny, the populations will do very well. We'll come back to why the islands didn't have lizards naturally in a few minutes, but the fact is the Shaners had put lizards on lots of little islands, and the populations had existed for 10 or 15 years by the time I was reading those papers. Well, this gave me an idea. My suspicion, which turned out to be correct, is that the islands differed in vegetation. Some would be more vegetated like this one with small trees and thick vegetation. Others would be much scrubbier with only small bushes and narrow vegetation. Well, based on my doctoral work, I knew how over millions of years lizards adapted to differences in conditions. On broad surfaces, they had long legs. On short surfaces, they had narrow legs. And it occurred to me that this was essentially an experimental test of this idea. The Shaners had put lizards on islands with different vegetational characteristics. The prediction was that they should evolve differences in how long their legs are. Again, so here's the prediction. So I contacted Tom Shaner towards the end of my graduate career and said, has it occurred to you that you've set up an experiment in evolution? And Shaner said to me what I could only have hoped for in my wildest dreams, but never really expected. He said to me, that's a great idea. Why don't you come do a postdoc and find out if that's happened? You know, success, exactly what I was hoping for. And there's a lesson here for you young graduate students and so on. It never hurts to ask. And so there you go. So pretty soon I was out on these islands measuring the lizards myself. As again, the prediction that the islands did vary in vegetation, and the prediction was that the lizard populations on the different islands, their leg length would vary in relationship to the size of the surfaces they were using on the different islands. So here I am on the islands. I would go in my little boat, take a chair with me, capture the lizards. Back then, our methods are very crude. I would just sit there and measure the lizards. Here I have what's called an ocular micrometer, where I'm looking at their toe pads, counting their lamellae. I would have a little ruler and measure how long their legs are, catch the lizards, throw them back, and so on. And here's what we found. And we found exactly what we predicted. Now, for some reason, we plotted the graph in the odd way. I should be perch diameter on the x-axis. But what this shows you that on islands where they were perching on broad surfaces, their legs were long, whereas on islands on which they perched on narrow surfaces, they had shorter legs. Exactly the prediction we had made. What this showed is over the period of about 15 years, which is about at most 30 lizard generations, they had evolved exactly as we had predicted. An experimental test demonstrating the relationship between perch diameter and limb length. Very exciting result. So we, I should tell you that this paper was published in the journal Nature in 1997. And we were in the Bahamas at the time, but Nature, as you may or may not know, every week puts out a press release hyping a few of their papers. And they decided to hype our paper. And here's what they wrote. This may be among the most important work in evolutionary studies since Darwin studied the diversity of finches on the Galapagos Islands during the voyage of the Beagle. Well, it was a nice study, but this was way over the top. What could I say? Now, we were actually in the Bahamas at the time doing follow-up studies, and we were staying in a little hotel, just before I left. Remember, this is the 1990s, not like today with the internet and so on. Anyway, at the last minute, I decided to change the message on my voicemail, basically saying, giving the number of the hotel I was staying at, just in case anyone needed to reach me. And so here's the hotel. The rooms we had did not have telephones. If you can see right here, this is the manager's office, the only telephone the place had. One day I come back from the field and the manager is waiting for me and he says, "The New York Times wants to talk to you." And the next day, ABC News. And the day after that, it was the Boston Globe and USA Today. The world's media went crazy based on this press release. And the owner of the hotel was completely perplexed. He thought I was who would come to the Bahamas to chase lizards? I thought I was kind of crazy, and my colleagues, and yet the world's media was beating down his door and tying up his telephone line to get in touch with me. So we got lots of publicity from the paper, and I'm probably, my career owes a great debt to the publicity that was generated by that. However, when I would give talks like this about this work, there was very often a question. Someone would raise their hand and ask this question. Usually it was a pesky botanist. And they would basically ask, how do you know that the differences in leg length are the result of genetic change? Maybe it's phenotypic plasticity. Maybe lizards that grow up using broad surfaces simply develop longer legs, whereas those that grow up using narrow surfaces use shorter legs. And how do you know it's not phenotypic plasticity? Well, this sounded crazy to me, but I kept on getting this question. I should tell you, so I kept getting this question. So finally I decided I needed to look into the literature on the effect of diameter that is used by a lizard as it grows up, diameter of the surface, on how long their legs grow. Well, it turned out that that literature was easy to look up because there was no research on it whatsoever. There was nothing in the literature. There was, however, a related set of studies that had some relevance. And I have to say that this research I'm about to tell you about strikes me as amongst the craziest studies I have ever seen. Apologies if any of you have done this sort of research, but it was often done on laboratory mice on rats, and they were looking at the role of exercise in bone development. And so, for example, they would take mice and have them run on a wheel for 10 hours a day, and then the control group wouldn't get a wheel. So there would be a bunch of slug mice, sluggish mice that didn't get to move around. And the question was, and they were doing this as they were growing up. And the question was, at the end of the study, did the mice that exercised, were their limb bones any different? Or the same experiment was done with rats, in which rats were forced to swim in a bathtub for about 4 hours every day. And again, the control group didn't have to do this. Or there were chickens that were put on a treadmill and made to run for hours every day. Now, you're probably thinking, that's not a chicken, that's a mouse. Doesn't Losos know anything about zoology? Well, the fact is, if you try to Google chicken treadmill, you won't get any pictures. So I'm using the mouse on the treadmill. However, I gave this talk a year or two ago, and after it, someone sent me this picture to show that chickens do use exercise machines. Anyway, the research from these studies was pretty consistent in that the animals that exercise more did show differences in their limb bones to those that didn't. But those differences were in how thick the bones were. It turns out, and I didn't realize this at the time, the bone is a very dynamic substance. It lays down calcium, it reabsorbs it from bones. Bones change their shape, and a lot of these changes have to do with the stresses you put on the bone. You put more stress on a bone, say if you're lifting weight as a weightlifter, your body will deposit more calcium to make that bone stronger. However, critically, those differences are not in the length of the limb, but in its diameter, in its width. And the results that we had were on how long the lizard legs were. So most of this work does not show plasticity in length of limbs. But there was one particular notable exception, and this was an old study from the 1950s on human tennis players, male professional tennis players. And basically, this study, if you think about a professional tennis player, this is someone who has been smacking tennis balls around his or her entire life. And so, and critically, during their growth as a young boy or girl to an adult, they have been smacking those tennis balls around. And so if there's an effect, you might expect to, of that stress on limb length, you might expect to see it. And critically, you can use the tennis player's other arm as the control. So compare their serving arm to their non-serving arm. And so that's what the researchers did. And sure enough, they found that the serving arm of male tennis players was longer than the non-serving arm. That arm length is a phenotypically plastic trait, at least in humans, with regard to playing tennis. Now, there is another explanation, which is possible, which you may have occurred to you. Perhaps only asymmetric armed people become tennis players. Maybe you have to have, for whatever reason, a longer arm. We can't rule that out. But the most likely explanation is smacking a tennis ball all the time causes your arm to grow longer as you grow up. Well, so that indicates that phenotypic plasticity does occur in limb length, but it's a long way from professional tennis players to lizards using different sized perches. So what we needed to do was we did an experiment where we got baby brown anoles from Florida and raised them at the St. Louis Zoo in two conditions. This is one condition, the broad perch condition. This is a happy brown anole. This is how they like to sit in nature. Limbs spread out, you can see a little smile on his face. This is good. The other experimental treatment was on narrow surfaces. This is not a happy lizard. He's hunkered down. He has to hang on for dear life. Well, that was the experiment. Baby lizards growing up on the two different size dowels. And here's what we found. First, we knew that there was sexual dimorphism in limb length. And that's what we found. Males have longer limbs than females, hind limbs, relative to body size. But here's the effect of treatment, narrow versus broad. The lizards that grew up on the narrow surfaces had shorter limbs. Phenotypic plasticity does occur in limb length. We were so surprised that we did the experiment twice and got the same results both times. Other people have conducted similar experiments on this and other species and mostly gotten the same results. Interesting study just out of Sweden found this plasticity in one species and not another. So it seems that limb length is a plastic trait in brown anoles. Now, nonetheless, when we compared the amount of difference we found in this experiment, as indicated by this red line here, broad versus narrow, and compare it to our populations in Staniel Key, we can see that the amount of variation produced by plasticity is relatively small compared to that we saw among the islands in our study. And this is a pretty extreme difference in this experiment between a very narrow surface and a broad one. And even then we couldn't produce, this is as much plasticity that was produced. And so this suggests that the variation in the experimental populations in nature was not the result of plasticity, but probably had a major genetic component. Now it's taken us a long time. We are beginning, we and others, to get at the genetic basis of limb length variation. We're not there yet, but I think in the next few years we can directly test whether studies like this, we are seeing genetic change in limb length, but the plasticity experiments actually suggest that a large component of the variation is the result of genetic change. All right, with that, I will now move to Act 2 of this study, and that has to do with the role of interspecific competition. Do species of anoles compete with each other, and does that lead to differences in habitat use and subsequently to adaptive change? And so to do these experiments, we moved slightly to the south in the Bahamas from Staniel Key down to the island of Great Exuma. And we now add a second player to our study, not only the brown anole, but the green anole, Anoles smaragdinus, a close relative of the green anole in the United States, which is Anoles carolinensis. These species differ in a couple of ways. The brown anole is much more terrestrial than the green anole, which is usually found at eye level or above, and the green anole has shorter legs. It's not the shortest leg anole, but using narrower surfaces, being more arboreal, it has shorter legs than the brown anole. So our experiment was very simple. The idea was we would put these two species together on some islands to see if they compete with each other, and then put each species alone to see what they're like in the absence of competition. And so again, we worked on small islands that didn't have a species on them. And we asked these questions. Can the brown, again, these are islands that naturally were too small to have lizards. Can the brown anole survive on these islands as they did in the first study? Can the green anole, if you put green anoles on these small islands, can they survive? And then what happens if you put them together? So we'll start with, can brown anoles survive on these islands? And here's what happened. We introduced 5 lizards. 1 year after the introduction, all the populations have at least doubled in size, and some of them have truly exploded in population size. 3 years into the experiment, one population is struggling, although it did rebound, but the other 9 are doing very well. So the brown anoles, as in the previous study, do just fine. What about the green anoles? Here the story is a little different. In year 1, 8 out of 10 of the islands have at least doubled in population size, although none gets as big as the brown anoles. But 3 years in, 4 populations have gone extinct. Another 2 are down to a single individual, in both cases a male. And so it seems that the green anoles on many islands aren't doing so well. One island, they're doing very well. So one explanation for why green anoles aren't generally found on small islands is there seems to be some problem. We suspect it's too hot or dry on these small islands, but we're not sure what. But finally, do the brown and green anoles interact with each other? Is there any evidence for competition? Now, we had set up our experiment in which we had identified 15 islands, small empty islands, and we'd put them into groups of 3 islands that were similar. And in these 3 islands, we randomly gave one island brown anoles, 1 island green anoles, and 1 island both anoles. So those were the 3 treatments replicated across 5 blocks. And so what we're looking at here now is in each block, we're comparing the density of green anoles on the island which occurs alone, allopatric, versus the island in which occurs with the brown anole. And what we find is that in all 5 blocks, in the middle of the experiment, green anoles, when they occurred alone, had higher densities than when they were with the brown anole. You see this across the board. And so what this indicates is that the presence of the brown anole has a negative effect on the green anole. It seems that probably through competition for resources, the green anole doesn't do as well. So clear indication of interspecific interactions, probably competition. Now, so we had shown that competition does occur between these 2 species and could be related to why they differ in their use of the habitat. Finally, we'll get back to the question, though, you may have, why don't the brown anoles occur on small islands? We've now seen in 2 different places in the Bahamas that if you put brown anoles on islands, the populations do just fine. They become very, very dense. So why aren't they there naturally? Well, when you think about a question like that, what may come to mind is occasionally something really bad happens, something catastrophic that eliminates the lizards from the islands. And of course, when you're talking about the Caribbean, what you're talking about when you talk about a major catastrophe is you're thinking about a hurricane. And of course, my mentor, Tom Shaner, had this idea as an explanation, and they put that out in the first paper they published. But Shaner had the bad luck of starting to work in the Bahamas just when there was a natural lull in the frequency of hurricanes. And so they started working right around here, and there just at that time weren't many hurricanes. There's a natural cycle that goes on. Well, that natural cycle lasted for about 25 years, until the mid-1990s. And in fact, it lasted until 1995. And here we are in 1996, and Hurricane Lily is churning its way across Cuba. Our study site is right here. We were very excited about this because Lily seemed to be headed right to our islands where we had experiments. And finally, we could find out, what is the effect of hurricanes on islands? So we were excited about that. We were less excited about the fact that we were on the islands at the time. So nonetheless, there we were. every day we listened to the BBC, the Bahamas Broadcast Corporation. Every 3 hours, they would give us an update on the latitude and longitude where the hurricane was. And the bank gave us little maps so we could plot the course of the storm. And so we could see what it was doing. Well, as soon as Lily passed over Cuba, it took a hard turn to the east. And as you can see, it was going to head south of where we were in Georgetown. Oh, well. But then it turned right back and it was heading straight towards us. And every 3 hours, we put our little dot on the map. It was coming straight over. It scored a direct hit. The eye of the hurricane went over us. Here it is right after it's passed. Well, the effects of the storm were dramatic. Many trees were uprooted or snapped in half. We counted 17 yachts that were sunk in right where we were staying. And so they were major, major effects. Now, just by good fortune, we had just finished our surveys for the year when the hurricane hit. So we were in a very unusual situation. We could say the islands were like this 2 days before the hurricane, and here's what they're like 2 or 3 days after. So as soon as we could, we went back, we got our boat, which was in a tree at the time. I can't believe I didn't get a picture of this. Our boat had landed in a tree, a little motorboat. We took it out, we went back out to survey our islands. Now, one thing I didn't mention, here's where the study was in Great Exuma. We had study islands on both sides of Great Exuma, on the southwest and the northeast. And so Great Exuma was in between. Now, when hurricanes pass over islands, they do slow down, but Great Exuma is only 1 mile wide, and so that's not enough to really slow the speed down substantially. So the islands on the two sides should have been hit by the same speed of the hurricane. So first we went to the northeast, to the sheltered side, if you will. And what we found when looking around the islands, clearly something had happened. Branches were snapped off, leaves were stripped from trees. You could tell something had happened, but by the same token, it was a quantitative effect. There was damage, and the lizard populations, we recensused them, and they were down by 40% or by a third in Green Anoles from a few days previously. So clearly the hurricane had had an effect, but clearly it was just a quantitative one. Then we went to the other islands in the southwest. This was my favorite island. This is the one island in which Green Anoles were doing really well. They were everywhere. As you walk through this knee-high vegetation, Green Anoles would be jumping left and right like little grasshoppers thick in the vegetation. Well, this was the island when we got back to it. Initially, we thought we were in the wrong place. All of the bushes were gone. All that was left was scraggly rachycalis that grows between the limestone rocks. The vegetation basically gone. Here's another island. You can see all the vegetation's been stripped away. This tree had been about 3 meters high with a broad canopy, snapped in half, the tree's gone. Here's another island. This had been a big bush, 1.5 meters in size, quite large in circumference. It too was gone. So the islands had been devastated. It turned out that censusing the lizards was very easy. There were no lizards left on the islands. We had seen that, in fact, as Shaner had predicted, the reason these islands didn't have lizards, hurricanes come along every once in a while and sweep them away. Even though lizards can colonize across water, it doesn't occur rapidly, and so that's why most small islands don't have lizards. Now, the last question you may have is, why did we find lizards wiped away on one set of islands on the southwest, but not on the islands in the northeast? As I said, it's not differences in wind speed of the hurricane. However, what probably is that, well, there was one clue. We went to those islands where they were gone, and this is what we saw on the top of an island. That is a sea star, starfish as we used to call them. Now, those of you who have studied marine biology know that sea stars don't live on the top of islands. And the reason it was there, obviously, is that the islands were underwater. There was a big storm surge of about 5 meters that hit the island with great force, tearing stuff away, and then submerged the island underwater for probably 6 hours. And so that was the mechanism by which lizards were stripped off the islands. However, the storm surge hit the big island of Great Exuma, which is about 10 meters tall, and that stopped the storm surge, and there wasn't enough open water on the other side of the islands, other side of Great Exuma, to generate a storm surge where our islands lay. And so that was the mechanism by which lizards were removed from some islands and not others. Well, this ended our study on interspecific interactions. And so that brings us to the third act in our play, and that is the study of predation. And to do that, and particularly what role do predators play in anole habitat use and adaptation. Once again, we move now to the north of Bahamas, to the island of Abaco, basing our work out of the town of Marsh Harbor. And our last actor, I think, in this play is this lizard. This is a larger lizard than an anole. It's a species called a curly-tailed lizard for obvious reasons. It's larger, about maybe this big. It's very terrestrial. It doesn't climb into the bushes. Now, the question was, what effect would curly-tailed lizards have on the brown anoles? It was known that curly tails eat brown anoles. This is a photo by Tom Shaner, but we didn't know, is this an important ecological phenomenon that happens all the time? Or is it just something that happens once in a rare, in a blue moon, has no broad significance? Well, we set up the similar sorts of experiments. This time we found 12 islands that had brown anoles on them, and we added we randomly chose 6 islands to get curly tail lizards, and 6 islands did not have curly tail lizards. Now, I do need to tell you, curly tail lizards live on some of these islands, on the larger islands in the area. And we've observed them colonize islands naturally. And so we were just mimicking a natural process. We would take curly tails from one island and we would take them from the mainland, but we'd put them on little islands, and the nearest island with a curly tail lizard was 100 meters away. So we were not bringing in a species that doesn't belong there. We're just mimicking the natural process of colonization. Now, I want to stop my screen share for just a moment to show you a film that was made about this research that explains it better than I can. While I'm doing that, I'm going to close my email so it will stop dinging. I forgot to do that. All right, now let me share a different screen to show you this film made by the Howard Hughes Medical Institute. And before doing that, I want to tell you one thing about this film. They did a great job, but one thing is they insisted on calling the people working with me, they referred to them as Losos' team. They were not my team. These were my collaborators, my colleagues. And so it was a joint collaborative work. All right, here's the first part of the film explaining what we did.
Jonathan wondered if he could also create an experiment that would lead natural selection to favor longer legs. He found islands that already had anoles that lived primarily on the ground.
That's Jason Colby, University of Rhode Island.
His team caught and measured each lizard. Then he brought in a predator, a species of larger curly-tailed lizards that eat anoles.
Here's the bad boy of our experiment. This is the curly-tailed lizard. Now, what we did is we added them to half of our islands. These are very voracious predators. What we wanted to know was, what effect would this guy have on the anoles on the island?
Would the anoles evolve longer legs to run faster, or larger body size to defend themselves?
Excellent question. Well, let's go back to the PowerPoint and find out. Where is that PowerPoint? Nope, that's not it. Oops. 1 second, technical difficulties. Where is the PowerPoint? Ah. Oop, I gotta hit the share button. Sorry about that. Oh, this is unfortunate. Here we go, sorry. All right, here's what happened. We put the curly tail lizards and predators on some of the islands and not others. And here's what we see. The control islands without the curly tails, we get this big population boom. These are the babies being born. But almost immediately, there's a 50% reduction in the number of brown anoles. Presumably the curly tails are eating the babies in particular. And this 50% difference persists throughout the experiment. So clearly the curly tails do have an effect on the brown anole populations. Now, the brown anoles are no dummies, that they immediately start perching higher in the habitat in the presence of these terrestrial lizards. Interestingly, this perch shift increases throughout the experiment. We don't know why it goes up and up and up, but clearly they are shifting their habitat use to move higher. Unfortunately, after 2.5 years, things were going great, and then this happened, Hurricane Floyd. Our study site is right here. So the lizard populations were decimated. However, something different happened here than previous hurricanes. We went back a month later, and what we found was that the islands were covered with baby lizards. And the reason is that there had been eggs in the soil, even though the adults were washed away, the eggs in the soil and in the rocks hatched out, and the babies, and so there were babies, and the populations recovered. And that allowed us to restart the experiment, predation experiment part 2, a few years later. We did the same experiment, but this time, again, 12 islands, 6 of them got curly tails, 6 not. But this time, knowing that there would be effect, we did something different. And what we did is, before starting the experiment, we measured the brown anoles present on the island. We caught them, we measured how long their legs were, and then we gave each lizard a unique identifying mark by injecting this non-toxic rubbery substance called an elastomer into their limbs. So each lizard got a different pattern of color and which limbs were injected, similar to bird banding, if you will. A lime green in the left humerus, and the right femur component of the limbs got an orange color. So each lizard was individually identified. Then we could come back 6 months later, see who survived, and ask the question, does limb length affect survival on islands with curly tails and on islands without? This, of course, is measuring natural selection. So I think we'll skip the second movie in the name of time, but here are the results. As in the previous study, I'm graphing this differently, but in the presence of curly tails, the number of lizards using the ground plummeted. They were moving up into the bushes compared to the control populations that were staying on the ground. So the same habitat shift. Now we measured what is called a selection gradient, which basically measures the difference in this case, the hind limb length of the survivors versus the non-survivors. If survivors tend to have longer limbs, that would have a very positive selection gradient, say 1.0. Whereas if selection was favoring shorter limbs, it would have a negative selection gradient. And if selection wasn't operating, if survivors and non-survivors didn't differ in their hind limbs, then the selection gradient would be 0. So remember, we calculated this. We had 12 islands. We calculated the selection gradients on the survivors on each island. What we found was that in the absence of curly tails, there was essentially no selection. Limb length didn't matter. But in the presence of curly tail lizards, the lizards with longer legs were surviving. We had shown natural selection. Longer-legged lizards survived better in the presence of curly tails, but not in the absence. Well, that was a very exciting result in its own right. However, this was the early stages of the experiment, in the initial 6 months. We also expected, as I've shown this graph before, that through time, the lizards would move up into the vegetation to avoid the curly tails. And we know what happens as they move higher, that they start using narrower surfaces. And in fact, previous studies showed that in the presence of curly tails, when they were in the bushes, they were using much narrower surfaces. Well, as I've talked already, we know how lizards, anoles, adapt to using narrow surfaces. They evolve shorter legs. So our prediction was that in time, selection would reverse itself and start favoring shorter-legged lizards once they're up in the bushes full time. Well, to our surprise, in the second 6-month period of the experiment, that's exactly what we saw. Selection favoring shorter limb length. That selection had flipped in a 6-month period. Well, this was extraordinarily exciting. Now, moving forward, our expectation was that this isn't going to flip back and forth every time. Once the anoles are stuck up in the bushes, selection every time period, every generation will be for shorter limbs. And so we expected that selection to continue favoring shorter limbs. Would the lizards essentially turn into twig anoles? Well, you can probably guess what the answer is, and that is we didn't find out, because not one, but 2 hurricanes hit our study site in 2004, completely wiping out the experiment. Very sad. Once again, we were able to start the experiment a few years later. That was in 2009, we began again. Since then, we've been hit by 4 major hurricanes. So the experiment has been limping along, but we really haven't been able to get a long stretch, hurricane-free stretch. We hope someday to be able to finish this experiment, but we'll just have to see. Now, briefly, I want to tell you about another experiment we did to test the idea of founder effects. This was after these hurricanes had occurred. You're probably familiar with the idea made famous by Ernst Mayr. The idea is that when a population gets started by a small number of individuals, just randomly those individuals will have a a biased, a random set of variation from the population that might be different. So you can imagine if we only start with 4 beetles for 4 bugs for a new population, they might all be red. And so Meyer argued that this is an important factor in evolution. I've always wondered, could it be important for animals? Because presumably what happens is that one animal, a female, say, washes ashore on an island. So the opportunity for a founder effect could be great with anoles. Well, we had the opportunity after the hurricanes cleared off our islands. We had empty islands that we knew could support lizards. So we did an experiment where we created artificially a founder effect on 7 islands to see if we could see the founder effect in morphology and genetics, and would it persist, and how would it interact with natural selection. So I won't go through these questions. We've picked 7 tiny little islands, and we moved lizards from this island in the background where they lived in a forest on big trees, 7 tiny scrubby little islands. Each island got 2 lizards, a male and a female. Now I do need to point out that females at this time of year in the spring are gravid already. So there's probably extra genetics from the fathers that she's mated with. But we put these pair of lizards on 7 islands. Here's a view of that island again. That's Tom Shaner. There's Dave Spiller. There's some more of them. So our prediction, the adaptive prediction, is you move from a forest like this to an open area. They're going to be on narrow surfaces. They should evolve shorter legs. Well, first thing is the population. You might have thought 2 lizards, no way they'll survive. Well, this is what happened. All 7 populations did great. They expanded quickly, as we've seen in other studies. There was a founder effect. This is showing genetic variation. I'm not going to go through it in detail, but what this shows is right here is the average, is the gene frequencies of the founder populations. This is a principal coordinates analysis on microsatellites, and you can see the 7 founder islands have genetic differences from the source population, and those differences persist over 3 years. So the founder, and that's shown, the 7 islands are in different colors and they stay in the same place. In other words, the genetic differences that occurred as a founder effect persist over at least a few years. Now, there were some changes in our experimental technique. This is my favorite piece of equipment ever. It's a portable X-ray machine, no longer sitting out on the islands with a ruler measuring the hind legs. Now we brought the lizards back to our field lab and measured them with the X-ray machine and got these great images. And we could see all kinds of other things. Can you see what this is? That's a snail. It turns out these lizards eat lots of snails. They also, can you figure out what this is? That's a baby lizard. These guys are cannibals. We found a few instances of this. This is the large male. He's got a baby anole inside of him. Also, just to show you, this is a female. She's gravid. You can see how big the eggs are inside of her. We also found in some places something odd like this. There was one island where lots of the lizards had their forearms snipped in half. We're not sure if that was crabs or seabirds or what. Anyway, the X-rays are fascinating. They're much more accurate and so on. So we're measuring hind limbs in that way. We measured toe pads. This is Jason using a scanner to get better images of the toe pads. We also looked at their dewlaps. This was Manuel Leal's work. I'm not going to talk about that. Here's what we found. Hind limb length, these are again the founding populations. We see variation in relative hind limb length across the 7 islands. 0 is the average for the population they came from. Well, we predict that they will, there is this founder effect. We also predict that limbs will get shorter as they adapt to narrower vegetation. That's exactly what we found over 3 years. All 7 populations, they got shorter hind limbs as predicted. However, we note that statistically, the year effect shows that they're getting shorter legs, but there's also a leg effect. Here's the founder effect, the differences created by putting 2 lizards on each island. And for the most part, there's a signal of this 4 years later. The founder effect in limb length is still apparent, even though they're adapting to the islands. So this was a very interesting finding. Of course, this is only 4 years. You could say, well, it takes time for the founder effect to get washed away by natural selection. That could well be the case. Again, I should point out that these differences we found from beginning to end on these islands were substantially greater than we produced with plasticity, so they probably had a genetic basis. But once again, we weren't able to find out. This is Hurricane Irene, a year later, Hurricane Sandy, and that ended the founder effect experiment. All right. I'm going to very briefly give you 2 encores of recent work, and I will introduce this work by saying this is the work primarily of postdoctoral fellows in my lab. It's some very exciting work that is related to what has been going on here. The first was asking, what about natural selection on behavior? Do lizards respond, have behavioral differences, and does natural selection operate on it? This work was the brainchild of Oriol Lapiedra, who was a postdoc. He's now back in Spain. And he basically asked, is there variation in how lizards respond to predators? Is that variation consistent? Can natural selection operate on it? So here's the idea, that lizards may have different propensities. Some are more willing to run out and try to get food, and others are more cautious. They'll stay on the tree. And one could imagine that, so here's the variation in their propensity to forage, if you will, that when no curly-tailed lizards are around, when no predators, the ones that are more willing to get out there and forage quickly will probably do better. They'll get more food. On the other hand, in the presence of predators, such behavior may be a bad idea because you get nabbed more often by predators. And so selection might favor behavior that is more cautious, if you want to put that term on it. Well, Oriol decided to ask whether this actually occurs or not. And so we went to the Bahamas. He captured a whole bunch of lizards, the same place we've been working in Abaco, and he measured their behavior in the lab to see what they would do and whether there were consistent differences among individuals. And to do that, he put a lizard in a box. It's in a box, the door is closed. He then put in front of the lizard a curly tail. He can't get out of the box, but he can see that there's a predator. Then he takes away the predator and asks how long before the lizard comes out and how long does it sit on the ground in what would be a dangerous position, having seen a lizard, seen a predatory lizard. Well, what he found was that the lizards varied substantially in how much time before they came out, and I'm going to focus on how much time they spent exposed. And he did these trials repeatedly and found that the differences among individuals are consistent. So this is an individual level difference among that occurs. And then he asked, do these differences in behavioral tendencies affect survival on islands with predators and without? And so he put them on 8 islands, or rather he measured lizards on 8 islands and then put curly-tailed lizards, as we've done before, on 4 of these islands. And here's what he found. First, in the presence of predators, survival was lower. We've seen that before. And in the presence of predators, the anoles move higher in the vegetation. Again, the same result. But does this variation in their behavior affect their survival? Well, in the absence of curly tails, there was no selection. Survival did not vary as a function of their behavior. But in the presence of curly tails, it mattered a great deal that the lizards that tended to stay exposed longer had lower survival. So in fact, natural selection does operate on behavioral differences among individuals. Now, of course, this was a very exciting finding, and the question is, would we see evolutionary change in behavioral tendencies over time? And unfortunately, Hurricane Dorian, a horrible hurricane that was in the news last fall that devastated, it's very sad, devastated Marsh Harbor and Abaco, went right over our study site. However, Oriol and other colleagues went back there in March. It turns out that these populations, for the most part, are still there. And so we'll see whether there are long-term evolutionary consequences of these selection on behavior. Now, the second study, speaking of hurricanes, was led by Colin Donahue, a postdoc in the lab, and by Anthony Harrell at the Natural History Museum in Paris, with whom I've worked for quite a while. And now we move to the Turks and Caicos in the very southern part of, just south of the Bahamas, and we're focusing on a different species, Anoles scriptus. Here it is, very similar, convergent to the brown anole and very long legs and behaves very similar. And this work was conducted on 2 small islands. And these islands were overrun by introduced species, cats and rats and goats and so on. And a group, Fauna and Flora International, was going to come along to remove the invasive species to restore the islands. And so Colin had the great idea of going there and seeing how the lizards would respond as the vegetation recovered, as the predators were removed, and so on. And so Colin and Anthony went there in late summer of 2017 and measured the standard things we do on lizards, their morphology, their habitat use, and so on. And then they left. They left on September 4th, 2017. 4 days later, Hurricane Irma went right over the Turks and Caicos. And 2 weeks later, Hurricane Maria did the same thing. And so Colin realized there was a great opportunity to see what was the effect of the hurricane on these populations. He had gone out there, he had all these measurements. He hadn't marked the individuals because he wasn't planning to look at the effect of the hurricanes, but he knew what the population was like beforehand. And so the question he wanted to ask is, have the hurricanes acted as an agent of natural selection? Very likely there was a lot of mortality, but was it random with respect to their morphology, or were certain lizards surviving better as a result of their morphology? So he quickly packed his bags, and 6 weeks after the original visit, 3 weeks after the second hurricane, they went back there and they remeasured the morphology. And what they found, I think surprisingly, was that the survivors on the islands had larger toe pads on both islands than the population had beforehand. Now, there are other explanations, and I'd be happy to talk about them in the question and answers, but the most logical explanation is that toe pads allow them to survive better, that lizards with bigger toe pads can hang on better, and they survive the hurricane. They're able to hang on and survive. This seems to be the evidence for natural selection operating during a hurricane. Very exciting study. However, as I gave talks on this, after that was published, people would ask me a question, and the question was, I would point out that we have known for a long time that the anoles on the mainland of Central and South America have smaller toe pads than they do in the islands. And we had thought this was, there could be many explanations, but it suggested, could selection from hurricanes be responsible for the larger toe pads on the islands? Now, personally, I thought this idea was very unlikely, because even if hurricanes favor larger toe pads, well, there's a long time between hurricanes, and during those times, selection then should favor the non-hurricane smaller toe pads, and you would expect that natural selection would drive them back to that condition, because hurricanes are relatively infrequent. Well, Colin wanted to see if there was any evidence for this. I begged him not to do it. Don't waste your time. I said, it's just not going to work. But he moved forward. He measured toe pads, and the important thing is he collaborated with a meteorologist, Alex Kowaleski, and what they were able to do is to determine throughout the Caribbean how many hurricanes had hit various sites over the last 70 years. And so looking at brown anoles, for starters, they looked at 12 populations, and here they are, and the color indicates how many hurricanes have hit those populations over the last 70 years, I'm sorry. And what they found is that the toe pads were areas that were hit by more hurricanes have larger toe pads relative to body size. And they corrected for all kinds of other things, temperature and so on. The results still stand clear. This seems to suggest that hurricanes have a long-lasting impact, and the areas hit by hurricanes, you see larger toe pads. Interesting. But then they did that throughout the entire, excuse me, the Anoles radiation, 180 species. And again, they measured hurricane frequency, and what we see, now, we know that the mainland doesn't get very many hurricanes. I'll point that out, but there's a lot of variation throughout the Caribbean as well. And what we see is that, again, there is an effect of hurricane activity on toe pad size. This effect is highly significant, even if you don't include the mainland anoles, just the islands throughout the Caribbean. And again, phylogeny was considered in the analysis, was controlled for, as were things like climate, tree height, and so on. Nothing else seems to, this result comes through regardless. And so it does appear that there may be an effect of hurricanes having a long-lasting impact, that individuals who can survive hurricanes, that effect lasts through time and leads to this biogeographic observation. So a very exciting study suggesting that hurricanes may have long-lasting evolutionary effects. Well, I'm getting tired of hurricanes. And I will just conclude by saying, I never expected to do so much work to have found out the effect they have over short and now long-term periods on evolutionary trajectories. I am hoping, however, that the hurricanes might stop perhaps, so that we can follow some of these studies and get to the bottom of the effect of predators and so on. Well, thank you very much. I think I'll wrap up now. I'm sorry I've spoken a little long. Again, it's a pleasure to speak with you, and I would be happy to answer any questions that we have. Thank you.
Brilliant. Thank you, Jonathan. That was really fascinating and beautifully articulated. I have a few questions, but let's open the floor up to anyone else who raise your hands and ask Jonathan some questions. Anybody? Ruth?
Yeah, good afternoon. Actually, Timo and Ruth sitting behind the same computer. Thank you very much for a fantastic talk. With regards to your explanation for the differences in your first set of experiments, potentially explained by phenotypic plasticity, you set up an experiment in a zoo where you tested the potential effect of phenotypic plasticity. I was wondering, did you consider taking lizards from islands where they had different limb lengths and breeding them in a common garden context to test for phenotypic plasticity as well?
Yeah, that's a great question. And we didn't do that initially because those populations were small. And we felt that if we took enough individuals to do that experiment, we would, potentially devastate the populations. And so we figured, let's do this other work and let the experiments run their course, and we'll do that later. It turns out that was a big blunder, because sometime later we went back to Staniel Key, that being 15, maybe 20 years later, and the Staniel Key Islands had changed, that the islands had become much scrubbier, probably due to climate, and the populations, some of them were no longer there. And so we basically missed our chance to do that. In retrospect, maybe we should have, but we didn't. We wanted to follow the experiment on those islands, and so we didn't. But that would have been certainly worth doing, and certainly there's more work to be done on this plasticity to see how it really works, how this actually occurs, and what long-lasting consequences it has.
Thanks, Jonathan. Brian?
Hey, Jonathan, great talk. Thanks. Obviously, islands, in the context of natural experiments, islands are just fantastic resources. You get these nice isolated populations that allow you replicable studies, which is the real challenge. If you had advice for young grad students or something now looking for examples of natural experiments like this, are there any things in a herp context that jump out to you as being analogous to islands that might be like really valid or really useful things to pay attention to try and replicate type things like this? Well,
yes, islands, we're fortunate because they are, we call them test tubes of evolution, and they're bounded, and the lizards really can't go anywhere. And so that's why they're particularly useful. There are some situations in nature where populations, because of habitat differences, essentially get caught in habitat islands. And they're not, they're leakier than islands in oceans, but that's one place where sometimes situations like this happen. One particular example that people are now looking at is cities, how populations of all kinds of animals and plants are adapting to urban settings. And urban settings can be insular if they're surrounded by non-urban areas. And that's one way to do this. Another possibility to measure selection is even if you have a population that's not bounded by a border of some sort, well, you can still measure individuals and try to account for the fact that some are coming and going. And it requires much more intensive sampling, and it depends on having a species that's somewhat not very agile, that will tend to stay there. It's difficult, and we've tried that with not great success sometimes, but that's another alternative, is to use open populations. And most people who study natural selection are measuring open populations, but the problem is not only that your animals can leave and they're gone not because they died, but because they emigrated, but also other animals can come in and there's gene flow. so that it isn't ideal, but at least for measuring selection, it's one way of going about it. But it is tricky, no doubt about it. I do think this work on urban organisms is very exciting, that we have changed the environment in a possibly replicated way, and people are just now realizing that yes, species can adapt to these changes, and it could be very interesting.
Brilliant, thanks. Any other questions from anyone? Maybe I'll ask one in the meantime, Jonathan. You used those VIE markers, those color markers, to mark the individuals that you released, is that correct? Did you use that to monitor them, and were they quite easy to recapture? And did you notice any migration within the body of those markers?
So the VIEs are basically, it's a non-toxic rubbery product that was developed in fisheries. And so the way it works is you actually have two chemicals and you mix them together and they solidify. And so you inject them shortly after you mix them together while they're still liquid and can be, with a syringe, slip underneath the superficial layer of the skin. And then they harden up. And the problem with that in our context is that it hardens pretty quickly. And so particularly if you were out in the field doing it, you would have to keep them on ice and so on. So it didn't harden, so you could still use it. So it wasn't ideal. And we did have a number of lizards that the mark disappeared. And perhaps in a couple of cases, if you manipulated their femur, their limb muscles on their hind legs, you could see that the mark had slipped behind the muscle. I think the bigger cause of them disappearing was that a little bit of it had hardened enough that it left a little thread outside the body, and it just kind of worked its way back out. That's our guess. So I would say they worked okay. We ended up injecting them in 3 limb segments instead of 2 to give us some redundancy, that even if one mark was lost, we could still tell which lizard it was. More recently, however, people in the lab have switched to what are called, I can't remember what they're called, but they're little discs that are incredible. They're oval and incredibly thin, and you inject them with a syringe, again superficially, and then you can see them with a UV light through the translucent skin of the lizard. Those seem to work better, and they have little numbers on them, and so that's what we've.
I'll chat to you about that sometime. We're trying with some of our frog monitoring with the VIEs, and hasn't been too successful so far. So just interested to hear from a lizard perspective. No, great. Anybody else? Questions? Anybody? Well, I will fill up.
Hi.
Oh, who's that?
John, there's a question by James.
James.
Hi, Dr. Losos. That was a great talk.
Thank you.
I was wondering your insights on looking for kind of analogous taxa to Anoles. I think they've proven themselves to be a wealth of insight for evolutionary ecology. And I think other studies in other taxa trying to look for similar trends have fallen somewhat short in trying to find like a group of reptiles that seems as flexible or capable as Anoles. I'd love to hear your thoughts on other groups that you think would be advantageous to look at.
Well, so that's a great question, and I do feel very fortunate that I stumbled into Anoles just through the luck of who I got to meet growing up, really. Anoles have a couple of attributes that put together make them very good for this sort of study. First, they're an incredibly diverse group of lizards that are quite not only species rich, but have a lot of ecological and morphological variety. the fact that they've diversified on islands gives not on the islands in the Caribbean, they're among the major vertebrate groups there. And so they've become very diverse and very common. And the fact that there are multiple islands has allowed them to evolve, to compare evolution on these different islands. And they seem to have evolved very much in similar ways. And finally, some of the species, like the brown anole in particular, are incredibly abundant. And so they're easy to study in the field. They'll let you get within a few meters and watch them. You can catch them very easily. And so some of the species can be studied in great detail. And then you have this great evolutionary background of the diversity of them. So there are other sorts of organisms of various sorts that have these attributes. One that I was very interested early on are the Phelsuma lizards, the day geckos found mostly in the Indian Ocean, although you do have one species in southwestern Africa, that are very diverse. And there has been some work in recent years looking at them, and they apparently they show some of the patterns of anoles, but not others. I'm very interested in agamas, their diversity, the tree agamas and rock agamas in throughout Africa and Asia. There's been some great work. I would say they're not as diverse phenotypically or as anoles, but there is quite a bit of diversity. You don't get the big communities of them that you do with anoles, where you can have as many as a dozen species occurring together, but there are real opportunities there. Years ago, I spent a little time in South Africa looking at cordylids. They're certainly a fascinating group, a little harder to do the behavior and ecology on because they're shy. So it's a difficult question to find. It seems that many taxa are good for some things and not others. To find groups that are good for doing lots of different things, anoles are particularly good at that. And the chameleons in Madagascar certainly, but hard to study there. Crystal and her colleagues have done great work on the South African chameleons, fascinating group, and have found lots of things. So I think, the part of the problem is, one of the reasons I was very fortunate in studying anoles, I didn't realize it when I started, it was that there's this huge body of literature amassed over half a century now of research on anoles. And that makes it very easy to take anything you're interested in and study it in anoles, because we have the background to build on. Whereas if you wanted to pick some new group, you've got to get all the basics beforehand. And that takes a lot of work. So I think there probably are lots of other groups that would be fascinating to look into, but we just don't have it's a lot harder because you don't have the background. So I don't feel I've given you a great answer there. Certainly there are some very diverse groups of lizards in particular. That's what I know that one could look at. All right, I think I've said enough.
That was great. Thank you.
You're welcome.
Brian, is your hand still raised or re-raised? Great. Harel, do you have a question?
Thank you. The predators on these anoles, either on the islands or on mainland?
I'm sorry, I didn't hear most of that question. I just heard the end of it. Could you please repeat it?
Sure. Have you looked at other predators that may have influenced phenotypic plasticity in these anole lizards besides the curly-tailed lizard?
We haven't. That would be really interesting to look at. These islands, particularly the ones we look at, these tiny islands, don't have a lot of predators. Probably the biggest ones we suspect are various birds, but we haven't looked at them. It would be fascinating to do that. A related question is that the anoles on the mainland, Central and South America, are equally as diverse as the anoles on the islands, but their diversity is in different ways. And the mainland has many, many more predators. And for example, the mainland anoles are much warier. They're much less active. They're much more cryptic, and they have morphological differences. A reasonable hypothesis is that's due to the much greater predation pressure they face on the mainland. And that would be very interesting to look into. And we've been trying to do a little bit on that, but it's a hard hard question to get your hands around how actually to study that. But I think looking at other predators, both on the islands or on the mainland, would be very interesting.
Thanks.
You're welcome.
All right, Devin?
Hello? Sorry, can you hear me?
Yes.
Yes.
Go ahead.
Hi.
Okay, cool. Thanks. That was a really, really interesting talk. I was just wondering, have you demonstrated trade-offs between sexual selection and natural selection in things like, I don't know, dewlap size or even behavior in terms of sexual signaling and stuff like that? Have you shown that maybe there's a trade-off when there's a predator present, that dewlap is smaller or less colorful or something like that?
That's a great question, and I know that Oriol has measured, I don't know that he's measured color of dewlaps. I think he's measured the dewlap size, and I just don't know the answer to that. I don't think that, well, I don't know. It would be very fascinating to study that, and you could imagine the same sorts of experiments where you measure how much lizards display and whether those are individually consistent differences, and then whether that affects predation by the curly-tailed lizards. But we don't know. Now, there was, so mostly the answer to that is a great question, hasn't been done. What we did find, however, in one of the previous studies, this was Manuel Leal's work coming out of his lab, is that on islands with curly tails, the lizards bobbed their heads to a smaller amount, that just like agamas in Africa, the anoles will move their heads up and down. And Leal took a crew of students and they filmed the students, they filmed the lizards, and they filmed their head displays. And what they found on islands with predators, the amplitude they moved up and down was less. Now, whether that was a genetic change, we have no idea. It could very well have just been a behavioral plasticity, but it did suggest that they were modulating their behavior because of the presence of the curly tails, basically trying not to draw attraction to them. So anyway, I think it's a great question and would certainly repay further work, but we haven't measured that. I think Oriol measured it. I don't know the results.
Okay, thanks. That's very interesting. Thank you.
You're welcome.
Great. Thanks. We have a few messages via the chat line. So Nitya, if you want to ask your question now, and then Crystal has a question, but she can't raise her hand. So go ahead, Nitya.
Right. Hi, Jonathan. Thank you for a really inspiring talk. I wanted to ask your question slightly unrelated to your talk, but so sleeping sites in anoles and in many agamids are known to be very specialized. And how much of an influence do you think that the requirement for sleeping on, say, narrower perches or higher perches influences their morphology, or do you think it's just constrained by or a subset of the perches they can use while they're active?
I think that's a great question, and anoles, like many species that you're familiar with, such as chameleons and others, sleep in the vegetation for the most part, most species. They usually sleep on leaves or on narrow branches. The idea being that any predator coming to get them will shake those surfaces, they'll wake up and jump off. And there has been surprisingly little work comparing the sleep ecology, if you will, of different species. There's been one or two very nice studies, one by Michelle Johnson, but for the most part, we don't have a good idea how species differ, whether there are consistent differences among species in height, where they sleep, in diameter, and what sort of surface they sleep on, and how that might relate to their morphology, whether the differences, the morphological differences relate to sleeping sites. We just don't know. I think one of the reasons this hasn't been done is that it seems to me that most people, researchers who go to the Caribbean, as I did when I was young, say, well, I'll go out and collect data on sleep ecology while I'm doing my whatever project I'm doing there. And what everyone realizes is you have to sleep sometime, and you just don't have enough time to, it's a full-time project to get the sleep site data and to do it well. And as a result, lots of people start it and no one finishes the project. So I think it's a great study that remains to be done to collect sleep site data on many species, see how that relates to all kinds of things. Does it correlate with morphological differences? What kind of selection operates on that, and so on. So the answer is, it's a great question. We don't know. And I must say, this and several of the other questions highlight something that I always like to point out, is that anoles have been studied to an extraordinary degree, almost more than any other group of herps. I wrote a book 10 years ago that at that time had 1,400 references on anoles, and I would guess the number perhaps doubled in the last 10 years. Given that, it is amazing how many basic aspects of their biology we still don't know about, even for a few species, much less all 400. And so there's just a huge amount of information, even for a well-studied group like anoles, that remains to be learned. But anyway, that's a great subject, and someone should do their PhD dissertation on it.
Thank you so much, Jonathan. Thank you.
You're very welcome.
Great. Thanks. Crystal?
Yeah, okay. Thanks, Jonathan. As with everyone, thanks for the great talk. What I was wondering, is whether or not with anoles, do males perch higher in the bushes and trees than, or further towards the tips than females? And if they do, then I have a follow-up question.
You're talking about during the day, or a follow-up on the sleep question?
Not necessarily for sleep, just anytime. Are they using the vegetation high? Are they higher in the vegetation or on the tips?
Well, there's a lot of sexual variation, both in morphology, sexual dimorphism, and in habitat use. And it doesn't obscure not so great to obscure the differences among species, but it certainly occurs. It's not entirely consistent across species, but for the most part, most species, the males perch higher and on broader surfaces than the females. In part, that may be because the males are also larger than the females in most Caribbean species. So higher and broader as a general rule.
Okay, and so do you then see a different effect between males and females when you have them over generations on different sized perches? So when you, so do the females show less of an effect than the males?
You're talking about the island experiments?
Yeah.
Let me think about this for a second. I should know that off the top of my head. I don't think that there was a strong difference that we detected. You know, I can't remember, to be honest.
Well,
go ahead.
Yeah, no, I ask because with the chameleons, the females usually perch lower and more toward the center of the bush than the males. So the males are also up higher, and we see quite a difference between the ecomorphs of the males with the limb lengths and the toes, the chameleons, hand and feet size, but a lesser effect with the females. And the males are the ones who are up higher and out on the edges of the bushes. So we think it has something to do with that. And I was wondering if the anoles were similar.
That's a great question, and I just can't actually remember at the moment.
It's okay. We can talk later.
Okay, let me get back to you on that.
Great, thanks, Jonathan. We have one more question from John Measy. He's just going to type it because he doesn't have a microphone. So he's sending that through now. Is there any evidence of hurricanes as a colonization mechanism?
Great question. Direct evidence, no. However, there's every reason to believe that a hurricane could pick up a lizard and deposit it somewhere safely. Not always, but it could happen. And it is very curious that there is a species of anole on the island of Grand Cayman that clearly came from southern Jamaica. And there are many hurricane tracks that run from Jamaica to Grand Cayman. So it's very plausible that that's how the species got there. So I think it's quite possible that hurricanes are a means of moving the lizards around. And that one time when we were present when the hurricane occurred, when we went back on the water 2 days later, there was just an extraordinary amount of debris that had been thrown into the water. Now, we didn't actually find any lizards on the debris, but it's very possible. So I would guess that hurricanes likely are a means not only of wiping out populations, but moving them to new places. We don't have any direct evidence, but it's very possible, I think.
Great, thanks. Very fascinating stuff, all of it. I'm sure we could ask questions all night in the case of South Africa, but I think I'll leave it there. And if anyone has any questions, maybe email them through via Melissa, our secretary. I think that's been an excellent first webinar for the HAA. So thank you so much, Jonathan, on behalf of the HAA for your time this morning, giving us the presentation. We had really good attendance. We had about 76 people in total, I think. So that was really great for the first one. So yes, I think if everyone, and thanks everyone for their questions, you can give a virtual clap using your little reaction button, or an actual clap if you unmute. So really great, thank you.
Well, thank you, everyone. This has been a lot of fun, and thank you for all the nice comments on the chat. And please feel free to email me, and you can just Google me and find my email address. It's easy to find, but always happy to discuss anoles or lizards or anything else.
Brilliant, thanks, Jonathan. And then yeah, just to everyone, so we will be making this available on our new HAA YouTube channel. There'll be a link to the video, so you can share that on to anyone who may have missed the talk. And for anyone who is a non-HAA member, really great to have you join us. And if you're interested in joining as a member, please get hold of us through our various social media or to our secretary, and we'd be very happy to sign you up as a member and explain to you what that entails. So thank you very much, Crystal. I don't know if you want to end off, but thanks very much.
Okay, so it's never straightforward to unmute yourself. No, I just, thanks, Jonathan, so much for doing this for us. We really, really appreciate it. And I can see that most people have stayed online for the whole time. We still have the majority of participants. And by the comments in the chat, everyone seemed to love it. So thanks so much. And we will perhaps have you as a participant during one of our next webinars. I hope. Okay, do we end the meeting? Yeah, I think John can end the recording.
Thanks, everyone.